Hello everyone. In this class, we'll learn how to enumerate WBCs. The aim of this experiment is to determine the total leukocyte count, and the principle is the number of WBCs in a known volume of diluted blood is counted and the number of cells in undiluted blood is calculated and reported as number of white blood cells per cubic millimeter of whole blood. The apparatus which are required for this experiment is you require improved Newber's chamber, microscope, WBC diluting fluid which is also called as Turk's fluid. We require WBC pipette, cover slip, lancet and spirit swab. If you see the components of Turk's fluid and its functions, it contains glycylactic acid 1%, the function of which is to destroy RBC and makes the WBC nucleus very prominent. It also destroys RBCs, that is why RBCs are not seen. And it also contains junction violet or methylene blue. Junction violet is 0.3%, which is going to help in the staining of the WBC. It also contains distilled water, which will be osmotically lysing the RBCs, and it also acts as a solvent. Coming to the procedure, clean and dry the pipette, cover slip, Newber's chamber thoroughly first, then under aseptic precaution, prick the finger. Then wipe the first 1 or 2 drops of blood, then obtain a good sized drop of blood. Then draw the blood up to the mark of 0.5 in WBC pipette, during which you should avoid the air bubbles and suck the diluting fluid up to the mark 11, that is Turk's fluid. Then mix thoroughly, holding the pipette horizontally between the palms. Then discard 1 or 2 drops of the fluid from the stem of the pipette. Then charging. Charge the Newber's chamber by placing tip of pipette on the surface of the chamber, touching the edge of the cover slip at an angle of 45 degrees. And allow the diluted blood to flow under the cover slip by capillary action. Take care to avoid entry of air bubbles and flow of fluid into the gutters, which is called as overcharging, and recharge if air bubbles are present or if there is an overflow or over discharge. Then allow the cells to settle for 2 to 3 minutes. Then place the chamber on the stage of microscope. Then observe first under the low power or the scanner to ensure equal distribution of WBCs on the all corner squares. When you see under microscope under the scanner, you'll be able to see all the 4 squares, and when you see in the low power, that is in 10x, you'll be able to see one of the corner square which is containing 16 squares. And the WBCs are observed as a round dots and which are refractile in nature, and you have to differentiate them from the dust particles which have rugged margins and they are non-refractile. Then you have to count the WBCs in a zigzag manner like in this fashion, and you have to count the number of WBCs in all the 4 corner squares. So the N will be, the capital N will be small n1, n2, n3, n4, that is number of cells present in each corner square. Then calculation part. We know the volume of one corner square, that is 1 millimeter into 1 millimeter into 1 by 10 millimeter, that is the depth. So it will be 1 by 10 millimeter cube. So thus we are calculating or counting the WBCs in such 4 squares. So the volume of such 4 square will be 4 into 1 by 10, that is 2 by 5 millimeter cube. So 2 by 5 millimeter square contains n number of WBCs. Therefore 1 cubic millimeter contains 5 by 2 into n number of WBCs. And as we know that the blood will be diluted in the proportion of 1 in 20. So 1 cubic millimeter of undiluted blood contains 5 by 2 into n into 20, that is 50 into a number of cells. So that gives the number of cells in cubic millimeter of blood or microliter. The report has to be written in this manner, that is, the total leukocyte count of my blood is, for example, 5,000 cells per cubic millimeter of blood. What are the other uses of WBC pipette? The WBC pipette can be used for diluting the blood, much less for counting RBCs in cases of severe anemia or for counting platelets. It can also be used for counting sperms and bacteria. What are the red cells not seen when counting the leukocytes? The red cells are not seen because they are hemolyzed by glacial acetic acid in the WBC diluting fluid, that is Sturg's fluid. The remnants of red cells membrane are faintly visible, and so they are called as ghost cells. Are WBCs are also lysed while doing WBC count? Yes, WBCs are also lysed. This is to destroy the plasma membrane uh, to expose the nucleus for staining. So while counting, we actually count the nucleus of WBCs and not WBC as a whole. What are the physiological and pathological variations in WBC count? Increase in the leukocyte count more than 11,000 is called as leukocytosis. It generally refers to an increase in neutrophils and hence synonymous with neutrophilia. Some of the causes for increased total leukocyte count physiologically, it can occur during diurnal variation. The count is lowest in the morning and rises during midday. It is highest in the evening and count is lowest after rest. There can be changes in after exercise. The count increased after strenuous muscular exercise, and it is probably due to the redistribution of the cells, and it becomes normal within 1 hour. Then count also is higher in newborns and infants. Count is increased in pregnancy and highest during the labor, and it also increases during menstruation. Pathologically, leukocytosis is seen in acute bacterial infections like boils, abscess, pneumonia, etc. It is also increased in allergic conditions like asthma, hay fever, and it is also increased during hemorrhage, burns, malignant diseases, and also uh, in these conditions. Decreased total leukocyte count is called as leukopenia. That is, leukopenia is the reduction in leukocyte count below the normal, and it is pathological and occurs in certain bacterial infections like typhoid and paratyphoid. Then also in cases of viral infections like influenza, measles, and smallpox, and also uh, some of the protozoan infections like malaria and Kala-Azar. What is leukopoiesis and where does it occur? Leukopoiesis, that is production of WBCs, occurs both in bone marrow and lymphatic tissues. The two major lines of WBCs are myeloid series, that is for granulocyte and monocytes, and another lymphoid series, that is for lymphocyte, which arise from pluripotent hemopoietic stem cells in the bone marrow. What is the lifespan of leukocyte? Normally, the lifespan of granulocyte is about 8 to 10 hours or few days, but lymphocytes recirculate between blood and lymphoid tissue and may survive for months. What is meant by the term agranulocytosis, leukocytosis, leukopenia, and leukemia? Agranulocytosis is nothing but decrease in the number of granulocytes. Leukocytosis is, as we discussed, it is increase in the WBC count beyond 11,000 per cubic millimeter of blood, irrespective of the type of the cells, whether they are granulocytes, monocytes, or leukocytes. Leukopenia is nothing but decrease in the number of white cells below 4,000 per cubic millimeter of blood. And leukemia is, it is a group of malignant neoplasm of WBC forming organs, that is bone marrow and lymphoid tissue. The total leukocyte count is generally above 40,000 to 50,000 per cubic millimeter of blood, and even few lakhs. And also there will be more number of immature cells in case of leukemia. This is in brief about counting of WBCs. Thank you.